ಎಲ್ಲ ಬ್ಲಾಕ್ ಏನೋ ಗೊತ್ತಾಗಿಲ್ಲ ನಂಗೆ. ಸ್ಕ್ರೀನ್ ಮೇಲೆ ಬ್ಲಾಕ್ ಎಲ್ಲಿದೆಯೋ ಇದಾ ಸೆಲ್ ಇದೆ ಇಲ್ಲೊಂದು ಅಷ್ಟೇ ಬಿಟ್ರೆ ದಿಸ್ ಇಸ್ ದ ಥಿಂಗ್ಸ್. ಆಮೇಲೆ ಐ ಥಿಂಕ್ ಐ ನೀಡ್ ಟು ಒನ್ ಮೋರ್ ಥಿಂಗ್. So how the life originated until the YouTube people will, one second. Okay, thank you. Probably everything is sorted out, then we will postpone. Screen is not clear, really guys. One second. One second guys. I don't know from when it started acting like this. The block, what you are seeing is like a chat box, so it should not shown to you guys. Okay, good afternoon Gopika and others, sorry for late joining. Okay, there is some technical issues, so that's why we couldn't meet in a proper time. Yes guys, whenever I'm taking a chat box, it's showing the blockings. Look. Good afternoon Rohit. Okay, so that means I can't properly see the this one chat now, because whenever I open the chat, it's started to showing like this. Okay, we'll see what happens. I'm launching the poll guys, so I'm waiting for YouTube to restore. Don't take tension about the these things, blocks and all the things. I will try to avoid the things, and I will off my this also. Nobody else. One second. Yep. Nobody else please on your camera, because otherwise it will block some more also. Okay, we'll see. We'll start the questions then. Okay, so we will minimize the use of chat box in Zoom today, because it will block the complete writings. Okay, the first question came. The first question is cell wall of prokaryotes is made of. Okay, the question is cell wall of prokaryotes is made up of. The options are chitin, cellulose, glucosamine, and mucopeptide. Okay, you can think and you can answer the question. But remember, who are in the Zoom, please answer in the Zoom poll only. Don't answer in the chat, because I can't open the chat. Okay, so I'm just launching the poll, and who are in the YouTube live can answer in the YouTube live chat. Okay, that will be a good thing. I don't know how to hide. Talk about them. Yeah, block in the below side is better than blocking. Don't think about occasional blockings. Okay, we will take care of that. The issue is like I can't disconnect and connect, because it will disrupt the YouTube live. So that's the thing. Okay, I will try to keep the chat box whenever I open into the sites only, so that won't make any problem, it seems. Or I will stop sharing one second. Okay. So I think I'm getting all the answers. We will see. Then I will close the poll. Then it's already 1 minute. Okay, I'm ending the poll and I'm sharing the results. 20% chosen option A, 27% chosen option B, 29% chosen option C, and 24% chosen option D. And I'm just waiting. Also in YouTube live, nobody gave the answer. Yes, so I'm stop sharing the results. Now it's clear, right? Good Lord. Okay, so everything all right now. Yeah, thank you. So I got some answers from the YouTube also. Now I'm like, okay, we can start it properly, right? So we'll solve this question, guys. So I'm not at all like, what are your polls? Okay, so 20% A, 27% B, 29% C, and 24% is almost like equal kind of votings. Okay, now guys, you guys can put the message in the chat box. Now it's clear, everything is sorted. Okay. So the question is simple. Most of you guys didn't think about what I'm asking. Okay, the question is cell wall of prokaryotes. Can you guys answer me directly? What is the composition? What is the name we gave for the cell wall of the prokaryote. Having it. Exactly, right. We call it as peptidoglycan, or we also gave the term murin. Okay. Yeah, exactly. Peptidoglycan or murin. Everyone know that. So we will try to find that. Chitin, no. Cellulose, glucosamine, mucopeptide. So we are like confused. There is no option like that. Okay. There is no option like none of the above also. Then what's the composition? There's like two compositions there, right? One is nag, sorry, nag and nam. Ha, ha. So nag means what? N-acetyl glucosamine and N-acetyl muramic acid. Exactly, right. There is like two compositions there. One is the N-acetyl glucosamine and N-acetyl muramic acid. Okay. So then you can remember one thing. Chitin is not the cell wall of prokaryote. You can compare it to the either fungi or exoskeleton of this arthropoda, all those things. Is there no full form? No. N is the where its linkage is there. Okay, linkage. Linking is the N, so it will direct a link to the nitrogen group. So we call it as N. Okay, so we remove the chitin, and cellulose is the actually cell wall of the plant. So you can remove this also. You may confuse with glucosamine and mucopeptide. Okay, mucopeptide, you can think about the muco or mucor, or you can think about all those things. Okay, peptide means protein only, a proteinous structure. So these are normally related to outside wall or it's inside of the fungi and all the things. So you can remove this also. You can think about N-acetyl glucosamine here only, right? So the correct answer is the N-acetyl glucosamine. Remember, in this type of question, the answer is not direct. You have to go for its composition. Okay, in the composition you will get the answer. So think about the things. Okay, so sometimes you won't get the questions directly, sorry, answers directly. You have to think a little bit about its composition also. Okay, so why it's a glucosamine? Because peptidoglycan or murin is the prokaryotic cell wall is made up of. Its composition is N-acetyl glucosamine, so that's why glucosamine is correct. Okay, we'll move on to the next question. Animal and plant cells differ in the, okay, they're asking animal and plant cells, how it will be different. Okay, movement, second option is growth, third option is nutrition, fourth option is respiration. Okay, I'm launching the poll. I don't think none of you will need any time, because you're studying plant and animal cells from so many years, right? Whenever like you are small only, you are started to differentiate between plants and animals in the environmental science textbook. Later you studied its structure of the cells and what will do all those things, right? Still you are getting confused. Okay, remember, sometimes you have to think about the things which only asked. I told you, right? Exactly. So many people are giving correct answers. Really? Think one more time, Gautam. So it's like the components and the organism itself may be different. Okay, lot of people got confused. Okay, that's a good thing. Manisha, think one more time. I'm just waiting from the YouTube also. I'm giving extra time because it's like easy question and everyone studied about it, so I'm giving extra time. Okay, so who are not right, try one more time. We still have time. And the good thing is I didn't posted anything today in the WhatsApp. Just want to check you guys remember about class timing or not. You guys remembered. I'm happy. Yes, Akhil got one answer. I'm waiting for Ashwin, Anu, Gopika. What are you guys doing? Try one time. Yeah. Then I can end the poll, it seems. Yes, I'm ending the poll. We'll see. And I'm sharing the results. 31% chosen option A, 13% chosen option B, 35% chosen option C, and 22% chosen option D. It's a tough match. right? Especially between A and C. We'll discuss what happens. So I told you the first and foremost thing you have to think about whenever anyone asks a question is about ha ha ha, good, good. So the thing is, you have to think about the question first, right? They're asking animal and plant cells, double quote I'm putting, okay, differ in there. Okay, they are not asking about the difference between plants and animals, or they are specifically asking about the cell, right? How the cells will be differ, the cells of the animal and the plant cells will be different. Okay, this is for one and one, this one, okay, options. The first thing about the movement, how the movement will be. Do you guys think our cells will do the movement? Yes, some cells may do the passive movement, some cells do the active movement. Passive movement means without spending any energy, like our RBC. RBC is circulating throughout our body or showing the movement, right? But it is not spending anything. The plasma is moving because of the pumping of the heart. The RBC also moving along with it, right? So second thing, what about WBC? WBC have the activity of movement. Yeah, we accept that. Do you guys really think all animals cells will show the things? Okay, exactly. If you take a muscle cell or heart cell or brain cell or any other cells, they won't show, right? In our body, only a few percent, like less than 1% of the cell is showing the movement. All the other cells are not at all showing. If you take other organisms, same thing will happen, right? So you guys don't think that animal will show the movement. Exactly, animal will show the movement, but animal cells, not all cells show the movement. Okay, not all cells show movement. You have to remember about it. Okay, not all cells show the movement. Okay, not all animal cells, because you guys confused with that, right? So not all cells, animal cells show the movement. Only as very few percentage of the cells may show, right? Plant cell don't show the movement. Okay, we understand. But if you consider this, there are some cells, it can show the partial kind of the movement. If you take an example of stomata will close and open, right? And if you take an example of a leaf will rise and fall, right? Those things will happen, but we can't consider for all. So plant cells normally confined to space. Some of the animal cells may differ, okay, but most of them are similar only. So movement is not the correct answer. And bending, all those things will be there, okay, but this is not like different, right? And what about growth? Okay, so how the growth will be become different thing? Okay, you guys thought about the cell wall probably. Okay, some of you choose an option 2. You probably think that plant will have the cell wall, so there is no like a confined kind of the growth. But animal cells don't have the cell wall, so it will have the movement. No, even though it may have the cell wall or don't have the cell wall, restriction will be there because of the cytoskeleton and other structures. And remember the things, even though it may have or not have the things, right? Cell wall or not having the cell wall, growth rate will be similar. The kind of growth it's taking is different maybe, okay, but it's a similar only. Okay, so this also not be that much major one. What about respiration? Okay, both of them will show which kind of respiration, aerobic respiration or anaerobic respiration. What do you guys think? Growth is known as the multiplying of the cells. Both of the things same happens, exactly. What about plants? Do plants breathe? Do plants breathe? Yes or no? Yes or no question. Okay. Do plants breathe? Yes, exactly. What they will breathe? They will take oxygen for respiration or carbon dioxide? What do you guys think? I know, I got you guys. Okay, I know. I exactly know that at least how the things will. Okay, I will on the poll. Okay. So, the correct answer, that is secondary. What plant take for respiration? Okay, I'm just asking about which gas it will take. Okay, so options will be A, O2, B, CO2, C, Both. D, none, because no respiration. Sorry, not this one. Exactly. O2 in morning, CO2 in night. Okay, I'm launching the poll, guys. Answer in the poll, and who are in the YouTube live chat can answer in the live chat. Okay, I'm just waiting for your answers. So question is this one. What plants take for the respiration? Option A, O2. Option B, CO2. Option C, both. Option D, O2 in the morning and CO2 in the night. Okay, so I'm just waiting for your answers. Okay, this is one of the dead easy question. So everyone can answer, I think, right? So it's not a tough question. And I think all of you guys, like almost 130 people are somewhere in Zoom and some 11 people in YouTube. I think everyone can answer that. Exactly. So I already got the answers. Another 30 seconds is there. I'm getting a kind of responses. So I'm like, it's not. It's a kind of funny. Okay, sorry for laughing, but it's kind of funny. Okay. Last 5 seconds. I'm just ending the poll in 5 seconds. Who are not yet answered yet, I'll just answer. Okay. I'm ending the poll. Okay, so I'm sharing the results. 34% chosen option A, 29% B, okay, 11% C, and 20% D. Okay. Exactly. Okay. So remember, guys. So now I'll tell you what happens in here. Okay. So yeah, plants will be there, and the plants is also a eukaryotic organism, right? Plants is also a eukaryotic organism. Yeah. So what happened? It will take both O2 and CO2 so regularly. Okay. But the purpose is completely different. Okay. So why it will take CO2? Because using the CO2 is the source of the carbon, right? CO2 is a source of the carbon, so it can produce the glucose from it, right? Glucose means C6H12O6. It need a source for the carbon. Hydrogen it had a water, and oxygen it have this CO2 only can give, right? So finally it need a carbon, so it's taking the CO2, but it can't respire. That means it can't take the CO2 to anything, because CO2 is almost useless, okay, for doing the respiration. Then what is plant will utilize for the respiration? It's exactly O2 only. Okay, so plant also take the oxygen inside, and oxygen means it's oxidative, right? Oxygen, oxidative, everything is same. It will oxidate the compounds. By oxidation of the compounds, you can break down the things and you will get the energy. Okay, so it may be a plant or it may be an animal. Okay, wherever the things you are thinking, the respiration is normally associated with the O2 only. Then another one question: does plant also? I told that it will intake the CO2. Sorry, O2. Does that means plant will remove CO2 also? Exactly. Plant also release the CO2. Okay, who told you it not? We always think like we are the animals and we will take the O2 and we will release the CO2. The plants are completely opposite. Plants will take the CO2 and it will release the O2. Okay, that's not true. Okay, it's completely false. But it's based on something. What happens is like plant will take CO2 from the outside and produce the glucose. It will take O2 from the outside and it will respire it. Okay. After the respiration, the CO2 will come, right? It normally don't release it outside. Instead, it will use the CO2 again for its photosynthesis process. Okay. And the same thing, when these things are happening, sometimes the O2 or something released, everything utilized inside only. Okay, CO2 and everything utilized inside only. But probably you guys heard that nighttime we shouldn't sleep under the tree, right? Why that happens? In the morning, due to the sunlight, photosynthesis will be happening. The plant will take the CO2 and utilize it in a photosynthesis. But in the night time, there is no photosynthesis, right? So what it have to do? Instead of using it in a photosynthesis process, it will release it into the air. Okay, that's why maybe the concentration of CO2 may be high in that particular area, and high CO2 concentration is toxic to us. That's why we may die, right? So that's why the people will tell, don't sleep under the tree at the night. Okay, so that's the reason. And remember, the plant takes CO2 only for photosynthesis. For respiration, it will take O2 only. Okay, so respiration, you can think about it in both of either in plants or animals, it will be oxygen only. Okay, then how will be the nutrition? The plants, Sachin, the plants, yeah, no, one second. The plants, it will be autotrophic, right? But the animals will be heterotrophic. So it will be different. Right, we spent a lot of time, guys. Okay, we don't get enough oxygen in the night. Exactly. So in the night time, you won't get any, depending upon the species. You will learn that in this one, plant physiology. Different types of plant species are there, C3, C4, and CAM, all those things. Okay, normally you won't get. During the this one morning hour only, you will get the oxygen, and plant never release oxygen for you guys. Okay, oxygen is just a byproduct. Okay, oxygen is the byproduct of the photosynthesis. It can't do anything. If it keeps the oxygen inside, it's a toxic. Okay, don't think that oxygen is like a super good gas. It will only give good things. Nothing like that. Okay, there is a, when it's in oxidation state, that oxygen will become superoxide. Okay, that superoxide is highly toxic. It will kill all the cells. So nobody wants to keep the oxygen inside, so it will release it. Okay, so there is a lot of stories out there, bro. Like once we go to the plant physiology, we'll go all those things. Okay. Bro, less surface area. No, like probably this based on the C3 and C4 nature of the plant. The normally grass will be the C4 and other things will be C3. Based on that, I think I'm not sure about it. The surface area relates to photosynthesis and respiration, not about releasing and more. I'm not sure, Chiran. Okay, so I think we will move on, guys. Any doubt? Any doubt? So. Like we are spending so much time in how they take O2. They have a lot of pores in them. Okay, they have stomata, they have cuticular space, lenticular space. Okay, from that they do have a lot of pores in them. We can't use removable oxygen for respiration. I didn't understand it. Really, right? So many things will come in plant physiology. Okay, I don't know. Probably tamarind tree will have this one, right? Lot of devils and all the things. That's why they told you. I think I'm not sure, bro. I'm not sure about tamarind tree releasing more CO2. Okay, that's why I told you. This is just a comedy. Okay, we'll move on, guys. Okay, so we'll go to the introduction of the eukaryotic cell. I like everything. So introduction of the things. When you compare prokaryotic cell, these eukaryotic cells are comparatively larger. Okay, why it will become so larger then? Because prokaryotic cell are smaller. I told you, if it's become larger, then what happens? It can't do the control complete surrounding nature like our India, right? But still eukaryotic cells are larger. What's the reason? Because exactly right, because this have compartment. Okay, it have a compartments. What is the meaning of compartments? For every function, it have a certain areas. Okay, probably you may see any district head or anything. We will have a separate area for like industrial area, right? What's the use of industrial area? In industrial area, a lot of industries will be there, a lot of affluent, all the things will be running. And another one thing, there will be like area like citizens will be there, and another will be the market, everything will be there, right? What happens if you mix everything, right? If you mix everything, you can't make it bigger. You can keep only a few shops, one industry, and like 10 or 20 houses, right? You can't make it expand because it will become too dirty because of either the industry or because of the market, anything, right? But if you have complete separate things or compartments, so you can do all the things. That's what happened in the this eukaryotic cells. Okay, almost like several billion years ago, eukaryotic cells originated. They've actually started a complete new era. Okay, because these are completely different, completely like more than 10 folds or 100 folds larger than the prokaryotic cells. Okay, exactly. Thank you, Jivan. So I will mention that also. And it also did the division of labor. A division of labor. That means every work will be done by a particular kind of the organelle. There is no need to think a single organelle, no need to think about all those things. Okay. So the changes will be happen here is also a protestant cells will be comparatively smaller, but it have all the works. It may contain extra one layer or not, right? It may contain sometimes the layer, right? It may be contain a silica shell, or it may be contain a pellicle, right? If you guys remember pellicle, right? Or it may don't have any cell wall or anything. That completely depends. If you take a fungal cells are completely different, right? Fungal cells are like it have a chitin kind of the cell wall, and sometimes this will become sinusoidal. That means a multiple nucleus will be there, right? If you take a plant and animal cell, that's again a completely different thing. Plant cell will have a proper cell wall and everything, proper structure and everything. Animal cells do not have any cell wall, but it's normally circular, but the shapes will be completely dependent upon the work what it's doing. Okay, all those things will matter. Okay, here is also shapes will differ depending upon the particular species. Okay, particular cell type and origin and the function also. Okay, so these are large and they do have the membrane bound organelle. Okay, they have the organelles which will be bounded by the membranes. A single membrane will be there. Okay, so we will move to the second. We will just see what are the different cell organelle present in here. Okay, so I may erase one or two things. Don't feel bad. So regularly it will have a cell membrane. Cell membrane means a plasma membrane. PM means plasma membrane. Okay, and it do have a cell wall. We are talking about a plant cell, and it have endomembrane system. What is the meaning of endomembrane system? We will learn that later. Okay, just remember it has some organelle, important organelle ER, Golgi apparatus, lysosomes and vacuoles, and it do have mitochondria for the energy, and it do have plastid, mostly chloroplast. Okay, and some other types of plastids also. We will see what are those types. And it have a ribosome, especially ATS, and it have a cytoskeleton. That means it have a structure or base inside of that how the cells are formed. Okay, this cilia and flagella may be present depending upon what kind of cell it is. Okay, so if you ask me an example, you can think about a bryophyta, right, or pteridophyta or algae. Those cells produce the sperm and eggs, right? They be learned about three types of the gametes. It may be a similar kind of the gametes, or different kinds of the gametes, or completely one larger and one smaller kind of the gametes, right? I think you guys remember about all those things. It will have a flagella, or some of the cells may also have a cilia. Okay, even though it's a very rare thing. So this will be there, and please remove this one. There is no centrosome and centrioles. Okay, plants do not contain any centrosomes and centriole. We will learn about what it will contain and how the process will be maintained later on. Okay, I'm just removing it. And it have a nucleus. Yeah. So it have a nucleus, and that nucleus will contain the genetic material DNA. Okay, and it will have some microbodies. What are those microbodies? It may be peroxisomes. Okay, peroxisomes, spirosomes. Okay, we will learn about all these things later. Okay, so later. Yeah, that's what we discussed, right, Venkat? So if you take any gametes in the bryophyta, pteridophyta, it do have a flagella, right? It do have a flagella. So don't think about regular cells. Regular cells won't have any flagella. So as you can see here, how can you differentiate a plant cell with the animal cell? Plant cell will be having a thick cell wall and a major, or you can see a largely visible vacuole. So it's normally cover most of its internal structure, exactly, and it have a prominent nucleus and endoplasmic reticulum, Golgi apparatus. We will go for in detail everything. Okay, and it may also have the chloroplast. They didn't put the proper color to the chloroplast. It's actually, look, now it's better. Okay, it may have the chloroplast. Don't say that it's looking like mitochondria. Sorry. Okay, yeah, so somewhat like this. So it will have the chloroplast. That's also major thing. Okay, what vacuole will contain? Vacuole will contain certain inorganic or organic substances. We will also learn about it. Okay, so you can just think about organic or inorganic substances, or like what we learned in prokaryotes, right? Inclusion body, similar. Vacuole is similar, but a little bit more complex. Okay, what is we will see, Manvita, where it is. We will learn about microtubules also. We will learn. Okay, so here you can see, right, microtubules. These microtubules are the one which are holding the cell. Okay, or it may help in the mitosis and meiosis process also. You can think these things as the ropes. Okay, how the ropes will be controlling our complete things, right? Our ropes will be jointed to one and another, and it's controlling the things, right? Exactly like that. So, microtubule is also a part of the cytoskeleton. Okay, so later come to the animal cell. How animal cells will be different, except it do not have any this one plastid. Okay, that's one major thing. And what are the other things? So if you take care, here you can see the centriole, right? It do have the centriole. Remember, plant cells don't have the centriole. Here it have the centriole. Okay, and when you compare it, the this one, mitochondria are comparatively larger. Okay, we'll come to that, Preetham. Okay, the gaps and all the things, these gaps and everything. Okay, and there is no cell wall, right? So these are the major things. How can you differentiate? Okay, cell wall is absent. There is no cell wall. These are not cell wall. Okay, these are not cell wall. These are others. They wanted to show it like a 3D diagram, but it's looking like a cell wall structure. Okay, it's not cell wall. This is just the cell membrane only. Okay, so, and they do not have any vacuole also. Remember, there is no vacuole. Remember, animal cell don't have any vacuole. Okay, cell membrane in tuna. I'm just I have to remove this also. Sorry, no cell wall. And yeah, so this our cell is 3D or 2D. Everything is 3D only. Okay, everything is like it's a three-dimensional structure, right? It's not a two-dimensional structure, any cell or anything. That's why they wanted to show it in that way only. But we normally, when in diagram and everything, we will only write the two-dimensional structure, not a three-dimensional, right? Yep. X, Y, Z, exactly. One question came. Okay, so we'll see. The outermost layer of the plant cell is, okay, the outermost layer in plant cell is a glycocalyx layer, cell membrane layer, plasma membrane layer, none of the above. I'm launching the poll. I don't think you guys need much time for this. Okay, answer it in 1 minute. I'm not giving any extra time, and I'm just waiting from the YouTube also to give the answer more quickly. Okay, quick guys, quick. Preetham, we'll see. It's just a like a representation only. Then we will go in detail about the each kind of the this one ER and what is this function, everything. Okay. Okay. I'm just waiting. No, there is no vacuole in animal cells. Normally, vacuoles will not be present in animal cells. Cells will have different kinds of the structures, but not the typical vacuole, what we will normally represent in the plant cells. Not that one. Venkat, really? Okay. I'm just ending the poll, and I got answers in the YouTube also. Okay. Correct, Manvita. I'm sharing the results. 32% chosen option A, 24% B, 17% C, and 27% chosen option D. Okay. So, and in YouTube, I got option D. Everyone gave the option D. Okay. So, again, come to the basic point. The question, you have to read the question properly. Okay. Exactly, Pawan. Okay, the outermost layer in plant cell. Okay, outermost layer. I'm not asking about innermost. If I write the plant cell like this, I have to make two layers. Outer layer will be the cell wall, and inner layer will be the plasma membrane, or you can call it as cell membrane, or you can call it as plasma lemma. All are same only. Okay, so which is inner? Inner is the plasma membrane. What is outer? Outer will be the cell wall. Okay, we will see glycocalyx layer. Glycocalyx layer is the outer. Exactly, it is the outer. But where? Outer layer in where? Outer layer in the bacteria, not in the plant cell. Not in the plant cell. It may present in some of the animals. That's secondary. We'll leave it. Okay, we are not taking care of that. So you can remove this cell membrane layer. That exactly means to plasma membrane. We know plasma membrane will be present inside. So you can remove it. That's also there. Semi-permeable membrane. And plasma membrane layer. Both are same. Cell membrane or plasma membrane. No, this is also wrong. Then what is the correct answer? The correct answer is none of the above. Remember, don't ever expect that the answer should be in the option. Okay, the answer may not be in the option. It may be like simply none of the above. Okay, so yeah, some of you guys thought about it and answered it correctly. Really, congratulations, guys. And who didn't thought about it. So next time, whenever you are choosing a particular question, so think about it one time. Okay, then it will be better. So you can choose the correct answers. So don't choose it simply by choosing what I don't know. That's what I'm saying. Okay, don't think simply. We normally, when we saw plasma membrane or cell membrane, so we will directly go there. Okay, without ever thinking the differentiation or anything. Okay, so hi Anand, don't sleep. So next is the cell membrane. So we will go for this one only. Okay. So the first one, cell membrane. So how we can call it as? We can also call it as the plasma membrane. Okay, and you can also call it as the plasma lemma, right? You can call it as the plasma lemma. So lemma means lemma or lamellae means layer. Okay, lemma or lamellae means layer. Plasma means the thing which contain the plasma or protoplasma. Plasma means fluid. Okay, that's why we call it as either cell membrane or plasma membrane or plasma lemma. Stewan actually first discovered it. Okay, Stewan actually discovered it, but he didn't mention it, how the structure and everything, because cell membrane is thin, very thin. Okay, so when you are just observing in a regular microscope, you never ever like come into contact with these things. Okay, you only just observe the animal, sorry, plant cell wall, but you normally don't observe the this one, okay, cell membrane. But when you are watching under the electron microscope, you can just watch something like this. Okay, something like this, not a clear cut differentiation between that. Okay, so you have to go deeper. That's why we don't have an electron microscopic image of the plasma membrane. Why? Because even in an electron microscope also, you can't see how the structures are made directly, right? So because this is not any like larger structure, it's very thin. Okay, so how it will be then? This will be the important and must layer. Okay, then how these are seen? Depending upon, no, nobody's seen the plasma membrane. Okay, nobody's seen the plasma membrane. Then how we know that all these are things are arranged like this only? Depending upon our experiments. Okay, in chemistry, how do you guys know that a particular compound will have that particular structure only? How do you guys tell that it have aromatic ring only, or having that alcohol group only, functional group only? That's also in the second position only. How do you guys will say that? How do you guys see that particular structure? How can you say that a benzene ring will be like this? You can never see it in a microscope, but you can do certain experiments, right? Based on that, probably you already reading about the experiments. While doing that experiments, you will come to know that, okay, in this particular, when we are doing reaction with this particular chemical, this functional group is replacing with some other. So we will get to know that, okay, probably this is also there and that one is also there. Then based on our knowledge, we will arrange it in a particular model, right? Exactly. in any of the case plasma membrane absent, no. Plasma membrane will be there, okay, in all the living systems except virus and virus, leave those things. So in all the other things, plasma membrane will be present, okay. Yeah, so based on that, how I will tell you, okay. So one second. Exactly, I will take this one. So how these things will be? Plasma membrane will be the outermost, sorry, not outermost, it's a must and important layer. It's mainly composed of the lipids and proteins and also the carbohydrates. Instead of using the carbohydrates, I can use the term like glyco proteins, or I can use some oligosaccharide or something. Doesn't matter. Okay. The lipids and protein composition will be high. When you go for the lipids, it contains the phospholipid layer. Okay, phospholipids. What is the difference between a regular lipid and a phospholipid? The phospholipid is the lipid which actually attached by a phosphate group. Okay, not that much of a different. Then how can I see all these things? Okay, so because when we separate that particular plasma membrane, okay, how can you separate? That's another question. Okay, yeah, we can separate by the lysis of the membrane. Okay, when you do the lysis, there is like several methods are there. That's not in the syllabus, we will leave it. Okay, when you do lysis or breaking down of the plasma membrane, we can separate the plasma membrane layer. Okay, when doing that, when you do the experiment, we will come to know that there is like different lipid and protein composition. So we came to know that, okay, it have a lipid and it have a protein and it do have some of the carbohydrate structure. Okay, then what we have to do? Then we will use certain chemical which can actually remove or separate these molecules. Okay, then we come to know that there are some interaction. Based on the interaction, we will come to know that this layer is actually amphipathic. Okay, what is the meaning of amphipathic? Can anybody answer? What is the membrane of amphipathic? It's actually amphipathic layer. Okay, what do you guys mean that? You know about the meaning of amphi because you already know about the amphibians. You can try. Yeah, exactly. This layer can having both polar and non-polar structures. Okay, so or you can simply you can put it in this way also. No. It have both things. That means hydrophilic and hydrophobic nature. Okay. Normally, what happens? A compound or a thing will be either hydrophilic and hydrophobic. Okay, normally it will be a hydrophilic or hydrophobic, either polar or non-polar, but these lipids actually having both these natures. Okay, it also show hydrophilic nature and it also show the hydrophobic nature. Okay, yeah, we will know that. We come to know that. Then when we treated this membrane with certain chemicals, we came to know that certain proteins will be easily removed. Okay, certain proteins will be easily removed. After removing of that proteins also, like if you again go for the analysis, you can still find some protein. Then we were told that some proteins are easily removed proteins. Okay, probably these proteins are in the periphery. Okay, that means in the outside of this membrane. Okay, we call those kind of proteins are peripheral proteins. Okay, peripheral proteins. That means these proteins can be easily removed. That means these proteins are attached to the membrane either by the van der Waals forces or through the electrostatic attraction. Okay, any of those means, not like too deep. It's like a smaller forces, right? Either electrostatic attraction or due to the van der Waals forces. So it's just attaching to the outer layer. So whenever you add certain chemicals, these proteins can be easily removed. Okay, so we call it as the peripheral proteins because it may be present in the outside. Okay, some of them are very strong. Okay, hard or we can't remove easily. Okay, so probably these are directly linked with the inside membrane, right? That means probably it's embedded. It's in the phospholipid bilayer. That's why it's not at all coming off. So we call it as integral proteins or intrinsic protein. Okay, integral protein or intrinsic protein. Okay, so when you go for further, Okay, when you go for some further analysis, there is actually another kind of protein is there. That's not in your syllabus. That's why I'm not mentioning it here. When you go for a little bit in depth, you also find that we also have the cholesterol. Okay, but the issue what happened is like when you have an intact cell and you put something which can remove the cholesterol, you won't get any cholesterol. All you only get only a very few amount of the cholesterol. But when you treat it outside or completely ruptured kind of the plasma membrane, and when you treat it with the same chemical, you got a lot of cholesterol. Then how do you decide? Probably cholesterol are mostly deposited on the inner layer of the plasma membrane. That means towards this side. Towards this side means we call it as the cytosolic or cytosol, or you can say cytoplasmic side. Okay, cytoplasmic side. Probably it will be situated towards the cytoplasmic side. So that's why when it's an intact cell is there, you won't get that much of cholesterol. Okay, but when you add certain chemical which can remove this carbohydrate, this will be removed easily when the cell is intact only. That means what? Probably this carbohydrate may be situated outside only. Carbohydrate, you can call it as like saccharides, because these will be oligosaccharides. So don't think about what is the meaning of oligo or anything. Think about it's a sugar. Okay, a saccharides will be there. Those saccharides will be present outside membrane. Okay, so this is how we constructed this particular layer. Nobody's seen it. Okay, remember, nobody's seen the particular layer. Okay, so this is the another one layer for the better understanding. So when you go for this, what I mean, I'm going to that. Okay, so based on all this knowledge what we had, we constructed a kind of the layers. Okay, I'm not going for the actual the Singer and Nicholson, a common layer how we predicted. Okay, our prediction will be this lipids will form a bilayer. Okay, lipid form a bilayer. What's the meaning of bilayer then? Bi means what? Bi means two. Layer means layer. Layer means layer only, right? There is nothing different. So as you can see, the phospholipid is forming a layer. One is the outer and another one is the inner. Okay. Then how they are arranging them? Okay. How they are arranging means this have a structure of a head with a tail. The head will be polar. Okay. Exactly. The polar head will normally situate or towards outside. It always protrude towards outside. When you go for non-polar tail, okay, non-polar tail, it normally goes towards inside. Okay, don't think about outside and inside which related to cell. The outside and inside which related to that membrane. Okay, as you can see, the phospholipid heads are towards this space also and against this space also, but the tails are present towards inside of the membrane. Okay, why it's happening like that? Because as you guys know, most of our structure, okay, whatever our structure may be or anything, will normally contain water based of things. Okay, so this particular phospholipid or lipid have to ensure or have to secure its hydrophobic groups away from the water, right? Because it's hydrophobic and it's non-polar, it should be away from the water. Exactly. Okay, it's protected from the aqueous environment. Exactly, Monita. That's why it will rotate itself. Okay, this is how, as probably you guys will see a kind of how can I say, ball-like structure what you normally observe in the oil and water structures. Okay, why it is happening? Because it's turning itself and forming a kind of circular layer or a kind of the layers where hydrophobic tails will be towards inside and hydrophilic heads will be towards outside. This is very, 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 very important. Remember, polar outside, non-polar inside. Okay, so it may be from this side or maybe from this side. Okay, and we already discussed and how this will be. And what do you guys think about the movement of this will be, phospholipid will be? Do you guys think this phospholipids are stagnant? This will be stay in one place without doing anything? No, that won't happen. Okay, what happens? These phospholipids can show the lateral movement. Lateral movement means the phospholipid which is present here can move towards here, can move away from here and it can come to here also. That means laterally it's moving. How it's happening? Because this is not a solid layer. Okay, remember, we got another one point. What is that point? This particular structure is not a solid layer. Exactly. Right. This is not a solid layer. So what happens? This phospholipid is actually moving laterally. So I'm writing here the phospholipid, what kind of movement it can show. This lipids can show lateral movement. Lateral movement it can show. Lateral movement means what? From one place to the another place. Okay. So what about the changing between one phase to the another phase, from above to below, below to above? Can it do? Exactly, it can do that movement also. That you can call it as transverse movement, or you can call it as like flipping or flopping. Okay, flip flop means what? The structure of this will rotate from this side. One second, from this side, and it can turn into here, and this one may turn into here. This particular movement is called flip flop. Okay, so this can also show the flip flop movement. Okay, flip flop movement. Okay, but what do you guys think? Is it easy? No, this is not easy. So this is one of the hardest. Flip flop movement actually takes several days. Okay, like 5 days, 10 days. Okay, depends, because it's like very hard, right? Because this hydrophilic head should turn out. Don't think about these structures in 2D. Okay, imagine that in a 3D structure, like something, how it will be if like thousands of people will be standing, their head will be above and their body will be below, right? Think about the same thing and just copy it and paste it on the other side also. Okay, then think how they will pass to from one side to the other side. Okay, just think about, imagine the things. Okay, you can't imagine that thing in a 2D, right? So it need a lot of time, because that polar head should pass through the non-polar structures and to the another layer. So it will show the flip-flop moment, but it takes a lot of time. But we told you there are like a lot of proteins will be there, right? So exactly, it have a proteins. So I'm writing it using some other colors. Okay. So when you write about the proteins, protein may be present in different forms. How it will be done? It may have the integral protein, or intrinsic protein, okay. Moment, both lateral and transverse. I will tell you. Okay. So yeah, this type of one, and another one is the peripheral protein or extrinsic protein. Proteins are iceberg in the middle of lipid ocean. Exactly, Manvita. Okay. So, exactly. So someone is asking like, what made us think about the lipids will show this kind of movement? Okay. So we have experiments on that, but you are bad. The experiments are not in the syllabus. Actually, you are good also. Otherwise, you could have remember about all those things. The experiment is simple. We take actually two cells, and one is a mice cell and another one is the human cell. Okay, and we gave different color kinds of the dyes for both of them. Okay, a lipid layer of the rat cell will contain a kind of the how can I say, a kind of the dye. Okay, phosphorescent dye, and animal contain different kind of the dye, and we will let it fuse. Okay, after for some time, like 20 minutes or 1 hour, if you observe it, the cells are not exactly like one side one color, another one color. It actually mixed the things. Every color is mixed. Okay, so that's why it made us think that these phospholipids are not actually confined to a single space. It's actually moving around itself. So it's not staying in a single place. Okay, similar experiment. There are like a lot of experiments are there, but that is not included. Okay. So second one is the integral and extrinsic proteins. Okay. So what are integral proteins? Integral proteins are proteins completely embedded within this phospholipid bilayer. Okay, and it's hard to remove. Okay. And what about this one? Peripheral protein. Peripheral proteins will be present outside. It may be directly linked, or it may be having some smaller force of attraction or anything. It will never itself embed within complete phospholipid layer. So either one will be the integral, that means completely present in inside, or it may be the peripheral, that may be present any one side of that. Then what do you think about the movement of these proteins then? These proteins sometime, or very few times, show the lateral movement. Okay, it may show the lateral movement, but it's like, how can I say, very low. Okay, very low speed and very slow movement. Okay, but remember, no flip flop. There is no flip flop moment. That means the channel protein or anything don't rotate back or rotate come. There is nothing will happen like that. There is no flip flop moment for the proteins which are present in the plasma membrane. Only the lipids can show the this one flip and flopping moment. Okay, I told you sometime. Now you guys know about these proteins and all the things. Okay, and what are the other things we have? We have some oligosaccharides, or you can simply think about carbohydrate or anything. Okay, this will be present outside of the things, and you can also find cholesterol, which is related to inside of the things. Okay, I'm repeating the same thing. And when I'm mentioning about flipping and flopping, I told you lipid needs lot of time. That means more than 10 days sometimes needed for the movement from one phase to the another phase, right? But our proteins are there. Proteins are actually helpful. Okay, how it will be helpful? There are some proteins which are called as like flipase and floppase. We are not mentioning it here. Okay, so there are some proteins are there inside this. These proteins actually help this phospholipid to move from one to another side. This protein is not rotating. Remember, the protein is not showing the flipping movement. The protein is helping this phospholipid to turn from one side to the another. Okay, and another side to another. And it's called flipase and the floppase. Okay, you don't need to remember those things, but remember only thing: the protein can't do the flipping and flopping movement, but it can help the this phospholipid to show the flipping and flopping movement. Okay, so these are the things, and these are the represented things. Okay, then actually two people will come. Those are Singer and Nicholson, as you can see here. Okay, these represented more kind of a model to this one. Okay, so the model name is fluid mosaic model. What's the meaning of mosaic? We already discussed about the mosaic in some way, right? Mosaic means a kind of structure which is made up of the different smaller pieces. Okay, so like this, if I write something, think about something, I don't know what I'm drawing. Okay, yeah, patches, you can say, like, by doing the patches you can create a shape, right? Or you can create a particular photo or anything. That's called a mosaic nature, and they called the plasma membrane or fluid in the fluid nature. That means first we know that it's not a solid, but we are not sure that what it is. Okay, they told it as a quasi-fluid in nature. Okay, what's the meaning of quasi-fluid then? Quasi-fluid is not a complete hard solid, or it's not a complete liquid. Okay, it's almost like a jelly. Okay, it's almost like a jelly. They told that this is not a solid, or it's not a liquid also, because liquid can't retain its shape and solid can't do all this movement and all the things. This is actually in between. This is a quasi-fluid. It's almost like a jelly. Okay, like a molten. Exactly. It's a jelly-like structure. Okay, they told that the plasma membrane have the fluidity. Okay, fluidity because of this quasi-fluid nature. It have a fluidity. That means flowing kind of the nature. That's what helped the plasma membrane to do all the things. Okay, to do the all, it may be expansion or it may be engulfing, all these things it will help, and they ensure about all the structure. Okay, so as you can see here, this is the updated model, 1972 probably they represented it. It's like a kind of a 3D structure, right? As you can see here, all the heads will be above and the tails will be below, right? And in between you can find the proteins, or what you normally seeing, there's an integral protein. Okay, and you can also see the peripheral protein. If you see outside, that means exoplasmic side. Singer and Nicholson. Singer and Nicholson. Okay, remember the scientist name, guys. So in sometimes they won't ask you like fluid mosaic model. They simply ask you about the model which is represented by the Singer and Nicholson model. Okay, so remember the scientists, these are Singer and Nicholson. So this model. So peripheral protein. If you observe, you can see a kind of structures here, right? These are glycolipids, or it may be the glycoproteins. Okay, what is the use of these glycoproteins and glycolipids? Some others may have some uses. We will see that what is the function and everything. Okay, so what's the use of these things? Why we need a carbohydrate on the plasma membrane? Because these carbohydrate works as a, like receptors. Okay, or this may also act like a sensors. Okay, you normally see that you will have A positive blood or B positive blood, AB positive blood. Then if I ask you what is A or what is B or what is B, some of you may answer like, sir, it's an antigen. Okay, then I may ask, what's the antigen then? Right, antigen, like two meanings are there relating to your blood group. What is the meaning of antigen? What do you mean by AB blood group? Right, that means these glycoprotein, it may be a glycoprotein. Okay, our name maybe we gave the name A glycoprotein. If this particular glycoprotein, A kind of a glycoprotein present on your RPC, then your blood type will be A. If a B is present, then it will be B. If both are present, that will be AB. If there is no this particular will be present, then it will be O. Okay, there are some other receptors also there. Okay, we will see that there are some bacteria will come and attach to your body, or it will infect us. We will say that not all this ones a virus will infect us. Only few virus can jump from the one animal to the another animal. Those only can infect us. We will say like that. Okay, how? For every pathogen to enter your body, it need a permission. Okay, yeah, that's what I'm mentioning. Okay, every pathogen to enter into your body, it need a permission from your body. Okay, or it can do some other things. Then where it will get the permission then? Okay, so the permission is like a key and all the things. Okay, first it should attach to some places. These glycoproteins are the attaching sites for them also. Some of these glycoproteins are attaching sites, and some of them are helping finding this receptor, either blood group or any transfusion and all the things. These receptors will help you recognize own cell from the other cell. This is like your face. Okay, how can you differentiate you from yourself? Yourself from any others? Because you can recognize your face. Exactly, these markers or receptors help the body to recognize its own cell with some other kinds of these cells. Okay, there are like lot of receptors are there. Exactly. Okay, so these are the things they mentioned about the things, and they also confirmed about the lateral and flip and flop movement of the lipids and lateral movements of the proteins also. Okay, so this is the more conserved and more proper model still what we are following today. Okay, so this is how the structure will be. Then we will learn about what are the different functions. Okay, because of this nature, this plasma membrane not allow all the things which are coming there. Okay, what it will do, it will not allow macromolecules, that's what Chirant, bro. Glycoprotein means glyco means sugar, protein means protein. Glycolipid means glyco means sugar, and lipid means the lipid. Okay, not allow macromolecules. Remember, it will not allow it because it will be comparatively larger. Okay, and these are selectively permeable, not semi-permeable, selectively permeable. That means even though it can allow smaller things, it will not allow everything. It's select the things. Okay, how it will be? Normally, non-polars will be allowed. Non-polar smaller liquids will be allowed. Okay, what about polar liquids then? Okay, polars are not allowed. Okay, so why it will be? Why and how it will be? So you can think about it. Okay, the outer head will be polar only, right? That's why what it will do, it normally stop polar things. Why it should pass the stop the polar things? Because the polar contains may water is a polar, and it may fill the complete body, and all the things may happen. Okay, so that may be the reason. So it will normally allow non-polar smaller substances, and it will normally don't allow polar substances. Okay, so the main reason is the polar compounds will be high in the hydrated nature. Okay, in the hydrated, it's not like a repulsion. Okay, polar compounds will be in a hydrated nature. That particular structure cannot pass through the plasma membrane. Okay, that's why it normally don't pass. But to pass essential polar compounds, it have a different channels and the carriers. Okay, it have the different kinds of the channels. These channels and carriers will pass down essential things. Will pass down essential things. As you can see here, one protein channel is there. And this protein channel, probably for any one polar compound, that polar compound only transfer through this particular channel, not from anything else. Okay, so there may be another one carrier is there. That carrier means it will carry, this protein will carry that particular compound from one side to the another side, but not other compounds. That means even though it can pass or do not pass, this will be specific to a certain compounds only, not all the compounds. Okay, we will see. So what are the function in that, we will see about it. Okay, I will write down all the things and I will explain one more time also. The first function is give boundary. Okay, it will give boundary to that particular cell. Okay, yeah, last slide. So first thing about first function of this is it will give boundary to a particular cell, right? One second, guys. So who are in YouTube, I can't access YouTube live chat. I'm just trying one more time. So we will see what happens. Okay. Yeah, now I can find, no issue. Yeah. So what happens? They will they will give a boundary to that. And what is the next one? They will make a selective permeable membrane. This is the selective permeable. That means it only allow the things which are meant to be, and it will remove the things, or it will not allow the things which are not meant to be. Okay. And what is the another function? Can anybody tell me the function? Any other functions? So it can also help in the ingestion. Exactly. It can also maintain shape. Then I will write this one. Help in capturing of food. That may be phagocytosis. Correct. Phagocytosis and phagocytosis. I think you guys remember what is the meaning of phagocytosis. Do you guys remember? Phagocytosis means drinking, right? Drinking. Cell is drinking. That means liquid, right? Exactly. It have the receptors which actually help in recognizing the cells, and exactly it also help in the excretion and secretion. Excretion, and all those things. Yeah, exactly. It also maybe help in the osmosis process, all those things. Then secretion means what? Secretion of the cell wall, secretion of the enzymes, secretion of the certain proteins, not proteins, sorry, certain molecules which are help to bind those things. Okay, all those things. Then any more characters or any functions which related to the plasma membrane? Anything else? I'm just waiting. Respiration, exchange. It can help in the exchange. Diffusion and osmosis, exactly. Exchange of material. Endocytosis, phagocytosis, phagocytosis, endocytosis. You can write all those things in here only. Transmission of impulses. Plasma membrane, not really do that, but it have carrier proteins. Yeah, you can mention that. It will have carrier and channel proteins. Molecules. Okay, it will have a carrier and channel protein. These actually do the things. When we are discussing about the transformation of the nerve signals from one end to another end, right? How it will happen? It will happen through the different concentration of the sodium and the potassium ion. Okay, then which one it will control, or where it's controlling? Everything happening in the plasma membrane. Okay, everything is the carrier or the channel protein which is present in the plasma membrane will help to do that. Okay. So one question came, just try to answer. In fluid mosaic model of plasma membrane, the proteins form a middle layer. Upper layer is polar and hydrophilic. Polar layer is hydrophobic, and phospholipids form a bimolecular layer. Okay, I'm relaunching the poll, and you can answer it. What is carrier and channel? Aha, we will see that, Chirant. One second. Later we will discuss. In next slides we are discussing about that. Okay. Okay. There is some mistake in the options. No. Yeah, I got it. One second, guys. Exchange of material. Exchange of material. No, don't confuse with that. There is some issues with that. I'm ending the poll, guys. It's already time. Okay. So I'm sharing the results. 11% chosen option A, 41% B, 13% C, and 35% D. And in YouTube, everyone chosen option 2. Okay. There is some problem is there. Sorry for that. A question is repeated. Okay. So we will see about the option. Proteins form a middle layer. No, proteins never form a middle layer. It may be present in between. Okay, proteins may be present as an integral protein, but it will never form a layer-like structure. Layer means it should be continuous. It's not continuous. It's just the in between here and there. Okay, and upper layer is polar. Yeah, if you take this one, and hydrophilic, they are saying. That means they are saying this particular layer is complete hydrophilic and polar. Okay. No, the upper layer and lower layer, if you differentiate this one as the upper layer and the lower layer, okay, this one is the upper layer as most of the people answered, and it's the lower layer. Okay, don't differentiate it somewhat like head is the upper layer, tail is the lower layer, head is the another lower, is the middle, something. Don't think like that. Okay. So if you remember, like upper layer of phospholipid and lower phospholipid layer, they are saying upper layer is polar and hydrophilic. Okay, no, this is not true. The complete layer is hydropathic, right? That means it acts like both polar and non-polar. If they say head is polar and hydrophilic, it's exactly correct. But if they say complete layer, it's not polar, it's hydropathic. Okay, so you can remove this. Polar layer is hydrophobic. No, polar layer is hydrophilic. The last option, phospholipids form a bimolecular layer. Exactly, phospholipids will form the bimolecular layer. This question is repeated. Like in the last class, I actually added the same question, but the third option will be here, and this option is here somewhere. Okay, this should be third. Look, I didn't made a change in the option also, right? So this should be here, and another should be there, and there is another one, extended sentence is also there. I just removed it and I put it here one more time. Okay, so think properly. The correct answer is phospholipid form a bimolecular layer. Why option 2 is wrong? Because upper layer means the complete phospholipid layer, not only the head. Okay, remember about it. So next one is type of the this one, transport. Okay, it will show a different kind of transport systems. Based on its nature, we normally differentiate into two types. One is the passive transport and active transport. How we differentiated it? We differentiated based on usage of energy. Okay, what's the meaning of usage of energy? The passive transport means no use of energy. No use of energy. Active transport means there is a use of ATP or GTP, anything, any energy molecule it's used. Okay, the plasma membrane show all the all two types of both the types of the transporting system. That means it will show the passive transport also, but it will also show the active transport also. And we will see, okay, how the different types of these things will happen and all the things. Okay, so we will learn about the transport system and everything in the tomorrow's class. Okay, so sorry, today's class is little lagged because of initial trouble and later little bit deroute and all the things in questions, right? So we could have finished another 2 slides at least transport, no problem. Okay, so this much only for today's class. From today tomorrow we will learn about the transport system and endomembrane system. No. 1 second, guys. If you have any doubt, you can ask now. Okay, I will come back to that action. Yeah, this is the structure which is given in the textbook, right? So this is the exact. That's it. Mohan, I didn't understand. Inorganic phosphate. Ippude inorganic phosphate means what is cholesterol? It's a kind of sterol. Okay, it's a kind of sterol molecule, which is actually an important component of the biological structures, and it's actually gives rise to several other compounds also. You can think about a kind of a lipid. You think about kind of a lipid. Sir, is transverse moment in phospholipid? Yes, transverse lipid is only in lipid, not in the protein. Erythrocyte means RBC. No, this one. Oh, next one. Previous one or next one you are telling? Oh, this one. No. This one is just a graphical representation. This is not the actual how it will be. Okay, this is the one how it will be. Even integral proteins also not like this. Okay, it is more like thread, something like that. You will learn it in the biomolecules chapter. So in biomolecules chapter, we will learn about the structure of the proteins. They will learn about the primary, secondary, tertiary, and quaternary structure of protein. They can't represent all those things here. That's why they given like a representation. So it's like simple representation you can think. So this is the alpha helix. There are like beta sheets will be there. So all these things they simply mentioned it like boulders. Thank you. Any more doubts, guys? You can ask if you have any. Is virus change morphology? No, virus normally won't change the morphology because the this one protein layers will be normally rigid. Okay, but as it changes the strain, if the strain is changed, then the morphology maybe become little bit different. That depends. It depends on the mutation. Yeah. Okay. So what we studied, we will go back one time. Okay, what we studied today? We study about this one and this one, right? It's not important. We will come back to the here. So as we go for the cell membrane, what we actually studied about here means the plasma membrane or the cell membrane is composed of proteins and lipids. Lipids has the important part in this, and lipids form a two-layer, or you can say a bilayer, right? That's made up of the phospholipids. If you take each layer and if you take a single phospholipid, the single phospholipid is made up of a polar head and the non-polar tail. Polar head normally always turn towards the outside, and the tail is normally turned towards the inside, right? So in between these phospholipid layer, you can find somewhere proteins. These proteins may be completely embedded in the phospholipid from travel from one side to the another side. Then we call it as the integral proteins. Okay, But this is not always case, because sometimes the proteins are not completely embedded. These are only present on the outside. Then we call it as the peripheral protein. Okay. In this peripheral protein, which are turned towards the exoplasmic space, that means outside of the cell, cytoplasm, may also have the glycolipids or glycoproteins, or you can simply call it as a carbohydrate. Okay. So what this will do, these are act like a receptor for the different antibodies or different other molecules. Okay. And when you go for inside to that membrane, in between you can also find cholesterol molecule. Cholesterol molecule also increase the fluidity of that particular plasma membrane, and it also help in this some other construction help. Okay. So these are the things. We don't know about the exact structure. In the earlier days we thought that it may be a solid and all the things. Later, when Singer and Nicholson gave their idea, they told that it's not a solid, it's not a liquid, it's a quasi-fluid. In between structure, the quasi-fluid nature is giving fluidic nature or fluid nature of plasma membrane, which is actually helping the plasma membrane to expand or do the phagocytosis or turn into different shapes and do certain mechanism. All those are based on the that particular quasi-fluid nature only. That's what they mentioned. So from there, we are actually using the fluid mosaic model of the plasma membrane only. Whatever picture we are having is on this one. No, yeah. So that's why we mentioned phospholipid bilayer. Okay, whenever we are mentioning about plasma membrane, how the plasma membrane is built up, plasma membrane is built up phospholipid bilayer. Okay, a single lipid layer is always single, so it's a bilayer, it's not a single layer. Okay, but considering both, we call it as a plasma membrane. It's just a component. Both the layers are component of plasma membrane. Yeah, they are saying in some way, somewhere there's in some way the corona is mutated or changed its strain. Okay, this they are working on. We have to see what happened. Why? Why did you got me, Chirant? I didn't understand. No need to apologize. No issue. Okay, guys, then I will close the YouTube session. If you have any doubt, please do put it in the Telegram. Okay. 1 second. I will come back to you, Akshay.